And our last speaker today, before we get to the panel session, is Young-Jin Lee from Iowa State University. And Young-Jin Lee has been a real leader in plant spatial metabolomics. So you'll see a lot of examples from actual plants in this talk. And he's been doing a lot of high resolution matrix assisted laser desorption ionization mass spec, some of the methods that Theodore talked about. And per Theodore's talk, also illustrated how Young-Jin Lee has been a big contributor to spatial metabolomics of plants in metaspace as well. So with that, Young-Jin, please.
Okay, thank you so much for giving me this opportunity to share our research. Do you see the screen? My screen, okay?
Yep. Yep.
Okay, so actually some of the things are hiding. Okay, so there are many different techniques we heard about in mass spec imaging. What I'll talk about is MOLDI imaging, which is more popular and commonly used because instruments more available. So basically in this technique, we are using laser. If you have any sample on the surface, we are using laser, especially UV laser that can focus down to a few micron size. So it's definitely single cell size. And then we get the mass spec data for each of the spots, and resting laser, we can get images. So what I'm going to talk about today is I'm going to show you a few quick plant applications. Then I'll talk about two new directions that we are trying. So our platform has been MOLDI OB trap. OB trap is a high resolution mass spectrometry that can measure the mass down to ppm level mass accuracy, and that is important for small molecules because there are so many possibilities. And also these instruments had MS capability so that we can directly determine their structural information from the tissues. Of course, FTICR, like Chris Anderton had, and also some other new mass spec imaging, like timsTOF might be also as good or even better, but this has been working fine for us so far. So before I talk about mass spec imaging of our plant data set, I would like to emphasize that some metabolites are not homogeneous even among the same cell types. So if you are doing cell culture work, that's not the information you can get. So here is an example to maize leaf cross sections. So as those plant people know, maize as a C4 plant, it had cranch anatomy. It had this bundle sheath cells. I'm not sure whether you can see my mouse. Let me change it to laser pointer. So it has two different photosynthetic cells, the bundle sheaths of this single cell layer, and then mass field outside. So when it did the mass spec imaging, we could see this compound called HMBOA glucose or DIMBOA glucose. They are insect repellent known in maize. They are known to be present in mesofield. But what we see is that they're not in all mesofield, but they are only present within a pair of bundle sheaths. So this is the kind of information you cannot get it unless you do the mass spec imaging directly from tissue, not from the cell culture. And here is another application to maize leaf cross section. So we are looking at two different chloroplast membrane lipids that you can see in negative mode. So SQDG, sulfur-quinobasyl diglycerols, are present in both bundle sheaths and mesofield, but PG3021, phosphatidylglycerol 3021, is mostly in mesofield, while PG32 is more on bundle shares. So basically, because of the one double bond difference, they have cellular heterogeneity. And this is B73. And when you looked at MO17 made, they have a very low abundance of PG31, but PG32 is more or less homogeneous. So basically, using the mass spec imaging, we can tell not only cellular heterogeneity directly from tissues, but also we can tell from genotypic difference. Then we looked at their hybrid B73 by MO17 and MO17 by B73. What we see is that they follow maternal inherited. So B73, MO17 has a similar images than B73, and MO17 by B73 is similar to MO17. I know it's difficult to see the images, so we this more of the quantitative information. So basically, this is a PG ratio in bundle sheets versus mass field per unit pixel area. Most of them, there are not much difference, but PG32, B73 and B73 by MO17 is more abundant in bundle sheets, while MO17 and MO17 by B73 is more or less homogeneous. So this is the kind of information you can get using the mass spec imaging technique. And another application that I would like to share is in application to Peronia as a lipid biomarker analysis. So this is a collaboration with Young-Jin at Iowa State University. So they have been working on Peronia, which is a membrane protein kinase important in signaling pathway of plant defense versus plant growth. And in shotgun lipidomics analysis, we found that there are some class of compound called arabidopsides are highly enriched in Peronia mutant. They are previously known as wounding response, but their biological functions are not well known. We confirmed they actually do have biological functions. For example, arabidopside A inhibits root growth. But then in terms of mass spec imaging, we have become interested whether we can get some subcellular localization information. So this is Arabidopsis leaf cryosection. And in this zoom in images, green color indicate pheophytin-A. Pheophytin-A is chlorophyll A after losing magnesium inside the mass spec. That's what is happening. And blue pixel is Arabidopside A. And when you look at all the pixels, what we find is that whenever there is Arabidopside A, there always the pheophytin A. So this data is obtained in 5 micron mass spec imaging data. Of course, 5 micron is not exactly enough to tell subcellular localization, but some chloroplast is bigger than 5 microns. From this co-localization, we can tell Arabidopside A is co-localized to chlorophyll A, that means chloroplast. So sometimes we can get also subcellular localization information. Then I want to talk about some of the new research directions that we are trying. One of them is on-tissue chemical derivatization for the untargeted metabolomic imaging. So one of the mass limitations in mass spec imaging in general is that there are not many molecules depending on the sampling size. If you are sampling down to a few micron size, there are not many molecules. And depending on especially ionization efficiency, you may not see many compounds. So that's a limitation. So some people have been adopting what we call on-target tissue derivatization, basically spraying some chemical reagent in small tiny droplets, then modify each of the metabolites with much higher ionization capabilities. So our idea is, how about adopting multiple chemical derivatization in consecutive tissue sections Then, for example, if you derivatized all the alcohol groups, amine groups, carboxylic groups, and carbonyl groups, then hopefully maybe you can get a much larger chemical information you can get with the mass spec imaging. So we have done some preliminary experiment. So in this case, we modify primary amine groups with coniferyl aldehyde, carboxylic acid with picolyl amine, and carbonyl groups with the Gerald division team. And we applied to two different genotypes. Again, MAGE is our typical model system, B73 and MO17, and also we applied to leaf and root cryosections. Combined, we can now see detect 656 new metabolite features that we could not see without derivatization. So out of this information, there are many things you can do. Like you can do principal component analysis to differentiate genotypes versus tissue types, and we can do some semi-quantitative changes about each of the metabolites. And of course, you can see many different compounds visualized. So one of the limitations is of this on-tissue chemical derivatization that people need to keep in mind is that maybe there might be some side reactions that could happen. And if you are not careful, you may misassign some of the compounds. And we see this especially in when we were developing boronic acid to derivatize bisphenol diol. So this bisphenol diol is supposed to react with the boronic acid, but then when we were screening matrix, so matrix is some organic compound that we spray on top of the tissue to enhance laser absorption. This compound, common organic matrix, 2,5-dihydrobenzoic acid, has a really good reaction with the boronic acid. That's what we want. That's not what we want. And what happens is this OH group of carboxylic acid and phenolic OH is right next to each other. And then just as like in bisphenol diol, the boronic acid reacts very well. Even for CHCA matrix, where hydroxyl group and COH is far away, they do react in very tiny reaction efficiencies with carboxylic acid. So what that means is we need to have a better understanding about all the possible side reactions. So we have done systematic studies for each of the chemical functionalities and against boronic acid. Of course, bisphenol diol has the highest reaction efficiency, about 95% yield, while also phenol and carboxylic acid has a pretty good efficiency, as we have seen in DHB. Even when they are meta-positioned, we have about a 50% efficiency, and carboxylic acid has some reaction yield, while many other chemical functional groups, they don't have reaction. So this is the kind of information that you need to keep in mind when you are applying chemical derivatization for untargeted analysis. But then more recently, we worked with Theodore Alexandrov group, and they now adopted this chemical modification to their meta space. So now we can use this chemical derivatization. And when we tried this, one of the limitations that you need to keep in mind is, so here is how it works. So if you have some chemical compound, let's say this is an underivatized data set, when they are derivatized, some of them are derivatized, but there might be some underivatized ones because reaction yield is not 100%. And those derivatized ones, then what will happen inside the meta space is they'll subtract the derivatization mass, and then they'll look for the possible annotation. But what happens is sometimes we may have some false annotation can happen in this derivatization data set. For example, of course, for this particular compound, of course, for the derivatized compound, we can get correct annotations. But this underivatized ones, metaspace may misunderstand and then subtract the derivatized mass. And if there is any match, then it may report false annotation. So how can we remove these possibilities? What we can do is, because this compound should be also present in underivatized data set, we are going to search underivatized data set against as if it is modified. And if it makes a hit, then those hit we can remove from derivatized data set. Through this analysis, we can remove false positives. So now, for the time remained, I'll talk about some new directions we are trying. This in vivo isotope labeling for the mass spec imaging. So many of you may know, metabolomics people have been using in vivo isotope labeling for many different purposes, like identifying some unknown compound or to find new metabolic pathways, and also metabolite flux analysis. But its application to mass spec imaging has been limited, and that's what we are trying to do in the next few years. So here is an example to developing route. If you think about developing route, there are two different nitrogen sources. One of them is coming from the seed, and there is some also nitrogen coming from outside as a nutrition. And in normal circumstances, there is no way you can tell the difference between the two nitrogens. But what we can do is, if you grow the major root in 15 nitrogen ammonium, then 14 nitrogen amino acid must be coming from the seed, and 15 nitrogen amino acid must be from nitrogen outside. So here is an example to major root cryosection. In the left two are 14 nitrogen amino acid grown in 15 nitrogen ammonium. So they are amino acid from the nitrogen from the seed, while the right two columns are 15 nitrogen amino acid. So they are newly synthesized from the nitrogen that came in from the outside. And by the way, this image was obtained by growing the major root about 10 to 11 centimeters, and then cryosection it 2 centimeters below the seed. That's where you can see genotypic difference. As you can see, leucine and isoleucine and glutamine, these 73 amino acids are mostly at the piece, while MO17 is more or less homogeneous. But then if you look at 15 nitrogen labeled amino acids, they are mostly from alanine, asparaginase, and glutamine. And of course, that's not surprising because they are major nitrogen carriers. But localization information is not changed between 14 nitrogen amino acid and 15 nitrogen amino acid. So basically, the plant cells cannot distinguish the nitrogen from the seed versus nutrition. And we can also calculate 15 nitrogen incorporation efficiency. And we compared between 4 different genotypes. And as you can see, glutamine, there is not much difference. That's not surprising because glutamine is the major nitrogen carrier. So they are very efficient in fixing the nitrogens. While alanine and asparaginase, we see high 15 nitrogen efficiency, incorporation efficiency for B73, while low efficiency for MO17, and hybrid showed somewhere in between. So what's happening is because B73 is low in nitrogens from the seed, as you actually have seen from this image, B73 has much lower. I lost my mouse. Okay. Much lower amino acid because the seed itself does not have much nitrogen, while M17 is high abundance. So B73, because it's nitrogen deficient, it is much efficiently incorporating 15 nitrogens from the outside. But then more recently, this the data and this is some research that are still in progress that we obtained in the summer. Dogweed, we grow dogweed in D2O because dogweed as an aquatic plant, they grow quite well, up to 50% D2O. And then when we get the images, this mass spectrum is actually obtained by fracturing the leaf and investigating inside. And we can see these compound chloroplast membrane lipid, MGDG, pheophethine-A, and DGDG. When they are labeled, you can see a lot of shift. And then if you do the imaging, then you can see for low labeled lipid, like +7 labeled, they are more on the tip, while highly labeled deuterium are more on newly developing tissue. So we can see some deuterium labeled along the development axis. So maybe I'm running out of time, so maybe I'll.
You have about 5 more. You have a couple more minutes.
Oh, okay, so I have time. Okay, good. And then the last data that I would like to show you is certain carbon labeling of major root tips. So this is the protocol that we adopted from Olon's group. So what they have done is they have done carbon flux analysis of major root tips. So basically, if you have growing major root, we cut it out at about 3 millimeter length, and then we grow in the media with glucose. But instead of 12 carbon glucose, we use 13 carbon glucose. And after the major root tip is growing about 1 centimeter, we cut it out another 3 millimeter at the tip. So they are totally new tissues. And then we did the imaging. And then we can see a lot of interesting things. For example, you can see this certain carbon citrate, but mostly at the very end of the root tips. So of course, many of you know citrate is important metabolites in PCA cycle. So maybe what that means is energy metabolism is most active at the very end of the root tips. While we also see 12 carbon citrate, so these are again new tissues. So this 12 carbon citrate must be from recycled material from the old tissues, and that is everywhere else. While lipid, in this case, certain carbon phosphatidyl acetic acid, they are actually more or less homogeneous along the developmental axis. And what we can do from this data set, first thing we did to identify the compound. So what we did is we compared 12 carbon data set and 13 carbon data set and tried to match them.
Let's do 1 more minute, Young-Jin.
Okay, we have very accurate mass. We can identify the compound, especially we are not only relying accurate mass, but also we are looking at how many carbons are labeled. So for example, adenine has 5 carbons, and we can see actually up to 5 labeling. So we can confidently identify the compound. If you see isotopal pattern, we can see something very interesting. For example, glutamine, glutamate, they have a very fast turnover time. So you don't have any 12 carbons, but some other metabolites like adenine or lysine, they do have a lot of 12 carbon metabolites. So what's happening is this 12 carbon metabolites, because they have a slow turnover time, we can still see it after 3 days. And I'll move over, we can see this pattern and similar patterns in lipid. We see 12 carbon short. So to less certain carbon abundant, very interesting, and that's probably something to do with fatty acid biosynthesis, adding 2 carbon at a time, and there may be 12 carbon acetyl ACP is hanging around with low turnover rate. And we see some interesting patterns, again, because of the different turnaround times. And I'll stop here. So our earlier work was supported by DOE, and on tissue chemical differentiation was funded by NSF. And that I'll stop here.
Excellent. Thank you very much. Do we have any questions? See one question in the Q&A. So I think this was earlier on in your talk. The question is, are the lipid IDs based on MS-MS or MS3 spectra?
So most of the times we don't do MS-MS. We do MS-MS if we really want to confirm the identification. We rarely do MS to the third. So the ID is not actually down to which side chain is which fatty acid.
Okay. Any other questions?
I'm trying to stop.
Oh, yep, one more question there. Okay, great work. Is parallel labeling of C and N possible?
Of course, it's possible. Yeah, we can use, for example, maybe 4 different growth conditions: 12 carbon, 14 nitrogen, 12 carbon, 13, 12 carbon, 15 nitrogen, 13 carbon, 15 nitrogen. You can do those kind of combination, of course.
Okay, excellent. I think that's it. I would like to thank all of our speakers one more time. Thank you for being a part and sharing your research with us and talking about spatial metabolomics, single cell metabolomics.